Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Let me introduce my team. There are Lia Yulia Budiarti, Erlanda Permata Derlin, Maulidia Khairada Amalia, Fahdyanoor, Shofia Hilwa Ihsanti, Yafi Fahmi, and we are from Medical Study Program, Faculty of Medicine and Health Science, Lambung Mangkurat University, Banjarmasin, Indonesia. In this opportunity, I want to deliver you our research about ability test of taro or Colocasia esculenta leaf extract to reduce coliform bacteria in river water. Let's start from the introduction. As we know, water is a major natural resource that meets human needs as a very valuable asset, and one of them is river water. We use water for human consumption and household use. However, the quality of river water in Indonesia is in a severely polluted state. The availability of this water often cannot be used directly due to the presence of microbial contaminants in water. The impact of contamination that are often found in the community are symptoms of digestive tract infection and diarrhea. They are often caused by coliform or Escherichia coli. So, to reducing coliform, we need disinfection, and the most commonly used method worldwide is chlorination. But chlorination produces disinfection byproducts in the form of halogens that can react with other organic substances so that they are toxic or genotoxic to animals and humans. And long-term chlorination needs to be considered, so we need phytochemical that can be an alternative in disinfecting water. One of amazing plan that can be investigated to address the issue mentioned before is Colocasia esculenta, or commonly referred to as taro. So there are many various phytochemical constituents are contained in the leaf extract of Colocasia esculenta, such as tannin, phenol, terpenoid, alkaloid, flavonoid, glycoside, and other. These phytocomponents have a high potential as an antimicrobial. Colocasia esculenta also is a species that thrives in the wetlands and may act as an alternative disinfectant. So basically, this study aims to analyze the ability of Colocasia esculenta leaf extract to reduce most probable number coliform in river water sample. The main material used in this study are Colocasia esculenta leaf extract that were sourced from Pemurus village in Banjarmasin city. So we do the determination. The leaves were cleaned first and then chopped into small pieces and dried in an oven at 60 degrees Celsius. Once dried, they were ground into a fine powder. That 100 grams of this powder was measured in 400 milliliters of ethanol for 3 days with continuous stirring. The mixture was then filtered through funnel cloth, and the ethanol was evaporated using a rotary vacuum evaporator at 60 degrees Celsius. And then the resulting extract maceration was stored in labeled airtight containers in a refrigerator at 4 degrees Celsius for future use. The extract was diluted as a treatment at concentration of 7.5 ml per liter, 15 ml per liter, 30, 60, and 90 ml per liter with distilled aqueous, which each extract is prepared for 3 repetitions. After filter the supernatant, then we analyze the total coliform using the most probable number method, include presumptive test, confirmation test, and total plate number. The data of this study were analyzed and obtained in a normal and homogeneous distribution. Furthermore, to assess the impact of the extract, the data were analyzed using the ANOVA test, followed by Duncan post-hoc test, with a confidence level of 95%. So here, the result from our observation on water sample from the Lulut River are known to contain coliform bacteria with MPN of 240 MPN per 100 milliliter, and total bacteria here is H.6 multiplied 10 to the power of 5 CFU per 100 milliliter sample that show the total coliform content in river water sample has exceeded the quality standard. And referring to health requirement regarding air and the result of statistical analysis, it showed that the effectiveness of Colocasia esculenta extract at 7.5 mg per liter to 30 mg per liter has not produced any effect according to health standard. When it's 60 mg per liter extract, it began to provide a reduction effect on coliform in water sample, and the 90 mg per liter extract produced an effectiveness that equivalent to chlorine. So it means increasing the concentration of the extract significantly reduces coliform colonization. The activity of Colocasia esculenta in inhibiting the growth of coliform bacteria is due to the role of antibacterial compounds. Phytoconstituents such as alkaloid, flavonoid, saponin, tannin, and terpenoid were identified as present in the extract. So, alkaloid can damage the integrity of the outer membrane and cytoplasmic membrane of bacteria. Flavonoid, which his main mechanism of action against Escherichia coli is inhibition of nucleic acid synthesis. Saponin, which have similar mechanism of action like flavonoid that make denaturation of cell membrane. And tannin can inhibit bacterial growth. Also, terpenoid, also causing damaged cell membrane. So, the result of the study indicate that Colocasia esculenta extract has antimicrobial properties in river water sample. However, the practicality of the extract for drinking water disinfection needs to be studied further, and this study is also limited to in vitro studies. The effectiveness and role of taro leaf extract as a disinfectant for disease prevention and control, it requires future research. So, in conclusion, based on MPN and TPC tests, Colocasia esculenta leaf extract has the potential to be developed as an alternative disinfectant in reducing coliform bacteria colonization in river water. So that's it. Thank you and wassalamualaikum warahmatullahi wabarakatuh.